In the previous lecture, we have completed consideration of the first stage of respiration, which is fresh air delivery into the lungs, or simply lung ventilation. Now we are going to consider all the rest stages of respiration. That include gases diffusion in the lungs, transport of gases by blood, gases diffusion in the tissues, and what is considered in course of biochemistry, this is tissue respiration. Let's begin. First, we need to consider the air composition. Actually, most important air for us is the alveolar air. But before consideration of its composition, it's necessary to know the composition of the atmospheric air and expired air, which all are different. As you remember from previous lecture, lung ventilation, at rest at least, provides almost constant composition of the alveolar air, with very little difference between composition after inspiration and after expiration. Inspired air, or simply atmospheric air, contains about 21% of oxygen and negligible amount of CO2, just about 0.03%. Then each portion of the inspired air adds some oxygen into alveolar air and decreases the content of CO2. The expired air, of course, differs depending on the depth of respiration intensity, but for resting state, average composition is about 16% of oxygen and about 4% of CO2. As a result, you can see oxygen is taken by the body and CO2 is released from the body. Now, what is the content of both oxygen and CO2 in the alveolar air? Let's put all the values into the table for two gases, oxygen and CO2. Atmospheric air, expired air, and the alveolar air. All these three types of air have different content. Atmospheric air exactly is considered to be about 20.93%. Of course, simplified, you may say 21%. Then CO2 usually is taken as 0.03. In some sources, you may find that there is a tendency to increase in CO2 even up to 400, but basically this is 0.03%. Expired air, as it was said already, its approximate value depend on each individual and state of the body, but it's about 16%, 16.3. And expired air CO2 content is about 4. And now the most important air for us. This air is exactly area where blood takes oxygen, and so oxygen content. Maybe it's unexpected, but the content of oxygen is minimal between all the types of the air, just about 14.5, between 14 to 15%. And CO2 also, maybe unexpectedly, is even higher than in expired air and makes about 5.5%. And exactly from this air, the blood takes oxygen, and into this air, the blood releases excess of CO2. And this constancy is the base for the constant oxygen of the arterial blood. Blood passes through lungs, and regardless of the inspiration moment or expiration, we have almost regular constant level of both oxygen and CO2 with minimal oscillations. Let's first consider why we have the difference between expired air content of oxygen and alveolar air. Why the expired air contains more oxygen than alveolar air. Let's recall what is the inspired and expired air. It differs from the alveolar air volume because only portion of the inspired air reaches the alveolar space, and part of this air remains in the respiratory airways. And when we expire correspondingly, the first portions go out of the airways, which is anatomic dead space. Why it's called dead space? Because there is no exchange and the air remains of the same composition, so it's simply atmospheric air. Let's see, the air, each tidal volume consists of the dead space volume, about 150 milliliters, and this air fills the airways. Then the remaining portion goes into alveolar space. Then expiration results in release first of this 150 milliliters of the dead space air, and only last portions come out of the alveolar space. And so, as a result, the expired air is 30% of the, approximately, for this given value, it's 30% of the tidal volume. The air which had no any changes, atmospheric air. And so addition of this atmospheric air to the alveolar air makes content of oxygen higher and CO2 content less. And it's easy if you consider this, but at first it can be unexpected. Then let's start to consider the diffusion of gases. Diffusion and gases follow the general laws of diffusion between air and fluid. And it's possible if there is a difference of partial pressure. Let's observe the situation when we have air and fluid, and gas is present in both air and fluid. Gas particles show, and this gas creates a certain level of the partial pressure of the gas in the air. But gas dissolved in fluid also has its partial pressure. For this picture, the amount of particles is approximately equal, and so there is no difference of pressures. So diffusion occurs, but particles from air may go to fluid and particles from fluid go to air equally. So net diffusion is absent. It's just exchange which doesn't change the content of particles. But if there is difference of pressure, like that, for example, pressure in the air is higher than pressure in the fluid. So there is, now we make higher content of air particles and pressure is higher therefore. So you can observe the net diffusion, and of course it goes down the gradient from higher area with higher pressure into area with less pressure. Gas diffusion goes predominantly into fluid, until the moment when pressures become equal again and diffusion occurs but without changes. Opposite case when fluid partial pressure is high. So let's add some more particles into fluid, and now pressure is high in the fluid. And so now net diffusion occurs in opposite direction, diffusion from fluid into air. Again, until the moment when both pressures become equal. So, and the first case, when pressure in the air is higher than in fluid, is our normal difference of partial pressure of oxygen between the alveolar air and venous blood that comes to the lungs. And so oxygen, according to all the physical rules, goes simply from alveolar air into blood. While the second situation is characteristic for the carbon dioxide. And difference of carbon dioxide pressures is directed from blood into alveolar air. So we can see that it's normal process of diffusion that occurs according to all laws of physics. Let's consider what is the partial pressure of gases. This pressure, partial, is proportional to the concentration in the air. Air is a mix of different gases, and each gas creates the partial pressure. And partial pressure depends on the content, concentration of the gas. It's according to Dalton's law. Of course, total pressure also is important. So, total pressure multiplied by concentration of gas in percent produces partial pressure. Generally, it's very easy. If gas consists 50%, a mix of gases has 50% of one gas and 50% of another gas, so that total pressure consists of the two halves, one half for one gas and half for another. Or let's make another sample calculation. If oxygen and CO2, just imagine that gases mix when CO2 is present 70% and oxygen 30%. It's just for example. Our real air, of course, is different. And Therefore, if total pressure, for example, let's take 100 millimeters mercury, so correspondingly, the partial pressure, according to the following previous equation, goes 30 millimeters partial oxygen pressure and 70 millimeters partial pressure of the carbon dioxide. This is just sample calculation that shows how we calculate the partial pressure of gases. Let's move to the real values for our air. Partial pressures of the oxygen and CO2 in the alveolar air. This is pressure that we are interested in most of all. So, mean level of the alveolar pressure actually is the atmospheric pressure. As you remember from the previous lecture, the alveolar pressure becomes decreased during inspiration and increased during expiration. For the resting inspirations, it's very little decrease, about 1.5 millimeter mercury less than atmospheric. And correspondingly for expiration, it's enough to create slightly above the atmospheric pressure so that air starts to move out. Standard value of the atmospheric pressure is considered to be 760 millimeters mercury. Of course, that differs and depends on how high the sea level we are located, but usually it's about like this in many places. And so, alveolar pressure, therefore, which is slightly higher during expiration, slightly less during inspiration, for average, is equal to the atmospheric pressure. And now let's consider composition of the alveolar air. Of course, composition means that it consists of the same gases as the atmospheric air, just a different percentage. And as you know about atmospheric air, and similarly for the alveolar air, number one component is nitrogen, which makes about 80% of the whole composition, gaseous composition. Then second place is occupied by oxygen, which is 14.5 average percentage in the alveolar air. Then we have CO2, about 5.5%. And finally, also we have the water vapor, which is in a gaseous state, so it's also a gas, additional gas that creates certain pressure. And as for this pressure, we know this pressure exactly because under normal conditions with regular standard atmospheric pressure and with 100% saturation of alveolar air with water vapors, it makes exactly known absolute value in millimeters mercury, 47 millimeters mercury. Because we have standard temperature, standard pressure, so the value is known. And if total pressure of all the components taken together makes 760 millimeters, we can calculate the pressure that goes only for gases, without water vapors. Standard value 760 minus 47 for water vapor makes only 713 for the remaining gases. And only from this value, 713, we can take percentage of other gases. You should not forget about the water vapor, which always should be subtracted from total pressure. And then we can calculate the pressure, partial pressure of oxygen and carbon dioxide in alveolar air. 713 is the total pressure. And so, with 14.5% of oxygen, this oxygen creates pressure which is about 100 millimeters mercury. Really slightly above, but it's negligible, so we can round it to 100. And 100 is easy value to memorize. Then PCO2 will be correspondingly less as percentages, 5.5. And it creates about 39 or approximately 40 millimeters mercury. And these are standard values for the alveolar air, 140 millimeters mercury, correspondingly for PO2, partial oxygen pressure, and PCO2. Diffusion of gases in the lungs. Now we can see how this diffusion goes easily under the laws of physics, because if there is difference of pressures, the gas will move down the gradient. And so let's put these differences. Diffusion occurs between the alveolar air and venous blood. The values for alveolar air we just have calculated now. For venous blood, it's necessary to memorize these values. PO2 and PCO2. For the alveolar air, these are 140. For venous blood, the pair of values is 40 and 46 millimeters mercury. And just the remaining column for the table is for gradient, difference. You see, difference between alveolar air and venous blood in PO2 makes 60 millimeters mercury. Difference for carbon dioxide is much less, actually 10 times less, 6 millimeters mercury. Direction of gradients is opposite. For oxygen, the difference is directed from alveolar air into venous blood. And for CO2, it's from blood into alveolar air. And as a result of this diffusion, venous blood becomes converted to arterial, and values of the venous blood, of arterial now blood, becomes exactly the same or almost the same as of alveolar air. Diffusion results in equilibration of PO2 and PCO2 of blood and alveolar air. And therefore, arterial blood values of PO2 and PCO2 are almost the same as of alveolar air. In reality, they are slightly less for PO2 and slightly higher for PCO2, just negligible difference, so we can simplify and say that arterial blood, PO2 and PCO2 are correspondingly 140 millimeters mercury. You understand that constantly new and new portions of fresh air come to the alveolar space, and therefore equilibration doesn't stop in the middle of values. It goes until full balancing of values to initial alveolar air values. Factors that affect diffusion of gases. These factors can be received from the following equation of diffusion. Diffusion is equal to, you see there are many indices here. K is the diffusion coefficient, just to make units balanced. So you can see here, first important value, difference of pressures. Of course, this is main thing, main driving force for the diffusion. Then S here stands for a square area, but let's consider delta P is directly proportionally determines the diffusion. Higher difference of pressure, clearly the diffusion becomes higher. Actually, all the parameters which are above will all be directly proportional, as the surface area available for diffusion in the lungs. And again, higher surface area produces higher diffusion. Actually, you may imagine, if we make deeper inspiration, the lungs are stretched stronger and alveolar surface increases in size, surface area becomes higher, and this provides, as a result, higher amount of oxygen diffusion. Then alpha, this is solubility coefficient for each gas, it's individual. And higher coefficient of solubility provides correspondingly higher diffusion with all the rest the same values of parameters present in this equation. For example, for oxygen and CO2, diffusion with even the same parameters will be greatly different because coefficient of solubility for oxygen is less and for CO2 is much higher, and approximately this is 24 times higher. So you remember that gradient for CO2 is only 6 as compared to oxygen. Oxygen gradient is 60 millimeters mercury. But nevertheless, the diffusion of CO2 is ended and completed even earlier than oxygen because solubility coefficient is great and CO2 easily passes through all the membranes and vascular walls. And last parameter, which is located in denominator, you see this is X, and X here stands for diffusion distance in the lungs. Diffusion distance is the distance of oxygen molecules to pass from alveolar air into blood and finally to red blood cells, to hemoglobin. This distance is proportional to the thickness of the alveolar wall. It doesn't take the full alveolar wall thickness, but anyway, it's proportional. Therefore, when alveolar walls become thicker due to some fibrosis or other processes, it changes the diffusion distance and becomes higher. And as a result, diffusion becomes less. You see, this is the only index that stands in the denominator of this equation. And so higher diffusion distance makes diffusion less, the only parameter that decreases diffusion. Diffusion capacity of the lungs. Among all the indices present in the previously considered equation, it's hard to calculate really. For example, calculation of the area in the lungs available for diffusion. It's not easy. We can approximately estimate this. Then diffusion distance. It's not equal even within one and the same alveolar wall. And so it's hardly calculated exactly. But to use this equation for calculation, we need to introduce specific index, diffusion capacity of the lungs, which is determined empirically. It's measured, and so we can use it for calculation. DC, diffusion capacity of lungs for oxygen, is defined as the volume of oxygen, in our case gas is oxygen, in milliliters, that diffuses through the membrane, through the lungs as a total, each minute for the partial pressure difference of 1 millimeter mercury. So if you look at this equation, you see the surface area remains, alpha solubility of course remains, diffusion distance, but delta P we make just 1. So therefore diffusion capacity practically becomes converted to this. Diffusion capacity of oxygen is all the rest components in equation just without delta P, difference of pressure, because difference of pressure now is 1 millimeter mercury. So if we know empirically how many milliliters of oxygen diffuse per each millimeter mercury of difference of pressure, to calculate the diffusion as a total, we need just to multiply it by one remaining parameter, difference of pressures, which is easily measured. So we see, therefore, diffusion of oxygen per minute can be calculated as follows. Diffusion equals diffusion capacity multiplied by difference of pressures. You see, milliliters per minute, and here balance of units is achieved by millimeters per minute divided by millimeter and then multiplied by millimeters. Therefore, we get diffusion simply how many milliliters pass each minute. This is diffusion as a whole. But basis for calculation is already known diffusion capacity. And how much this diffusion capacity is at rest. At rest, it makes about 25 milliliters per minute per each millimeter mercury of difference of pressure. It's necessary to remember. It can be slightly variable, not necessarily only 25. It can be 25, 30 or so, but for calculations, usually we take this standard value, 25. At rest, therefore, we can calculate how many milliliters or even liters of oxygen diffuse. Difference of pressure, as you remember, is 60 millimeters mercury between arterial pressure 100 and venous blood pressure 40, so 60. And then we multiply this value of diffusion capacity, 24 milliliters, by 60 millimeters mercury, and we get 1,500 milliliters per minute, or simply 1.5 liters each minute. This is the amount of oxygen that diffuses through the lungs from alveolar air into blood of the average person. Maximal diffusion capacity also is known. They say it's between 65 to 70 even. So let's say maximal is really 70 milliliters per minute. Of course, not any person may reach this value, but for humans it's possible for a trained person. And this increase, how does it occur? You see, it's not achieved due to difference of pressures, because pressure difference for diffusion capacity is always 1 millimeter. So for 1 millimeter, it doesn't make difference. Increase of diffusion capacity is achieved due to higher diffusion area, because we breathe deeper, and deeper inspiration stretches the lungs stronger. So diffusion area, surface area of the lungs increases. And with increase of alveolar size, we have decrease in thickness of alveolar walls, and therefore diffusion distance also decreases in the lungs. So look again at this equation of diffusion capacity. You see, surface area increases and diffusion distance decreases. As for alpha solubility coefficient, under normal conditions, it is standard. It can be increased when total pressure, atmospheric pressure, increases, so like in barometric chambers. But under normal conditions, it is standard value. So basic change of the diffusion capacity is achieved by higher surface area available for diffusion and less diffusion distance. Let's consider this diffusion area in more details. Look at the picture of the capillary network in the alveolar walls. You see how dense are capillaries and the lungs, and actually they take almost the whole surface of alveolus. And diffusion surface area is even wider than only the surface of alveolus itself, because you see capillaries have walls, and each side of this capillary in three-dimensional space represents the diffusion area. So total area of the lung capillaries is considered to be about 70 square meters for average, and with variability even up to 100 square meters. It's very great surface area available for diffusion. Then you can observe alveolar wall cross-section, and you see that it's very, very thin, and to the up and to the down direction from this, it's alveolar space. Layers of this alveolar wall: epithelial cells, and they are very, very thin, except for the areas where their nuclei are located. Then there is interstitial space, which is very little, extremely small. Just a little space between epithelial and endothelial cells. And the last layer is endothelial cells of capillary walls. And also they are very thin, except for the areas of their nuclei. And generally the cells are very thin, flat, and they represent very short distance for passing for oxygen molecules. Red blood cells are located inside, and they are very close to the walls, so oxygen goes easily through these three layers. Epithelial cells, interstitial, and endothelial cells, three layers. And oxygen should pass through this short distance. Average thickness is considered to be about 0.6 micrometers. It's maybe average, like in this place, but we can find even thinner area. Of course, in the areas of the nuclei, it's longer distance, but let's see. Here, for example, the distance is even smaller than usual. It's about 0.1 micrometers. And if oxygen molecule passes this very little distance, it immediately appears in red blood cell, which is close, adjacent to walls, and inside the red blood cells there is hemoglobin that binds oxygen. So in some areas molecules should pass longer distance, slightly longer, in some areas shorter, but here, for example, oxygen immediately appears inside the red blood cells. Before we consider the processes of transportation, we need to consider very important ratio: ventilation, perfusion. First, let's consider what is it itself, then we consider the significance of this ratio. First, how it's calculated. Ventilation-perfusion ratio calculated the following way. Amount of the alveolar ventilation, not minute, but alveolar ventilation. You see, we don't need to know how much air is spent for dead space. We need to know how much fresh air reaches the alveolar space. So it's clear the alveolar ventilation, which is estimated approximately for average about 4 liters per minute. Then the perfusion. The word perfusion means passing through, but exactly this passing through refers to the blood supply. Actually, perfusion is amount of blood supplied, and how much blood is supplied to the lungs each minute. Actually, the index is one and the same for any part of the circulation. You remember, this is cardiac output, amount of blood that passes through the circle each minute, and it passes through the lungs the same amount as through systemic circulation and so on. So lung perfusion is simply cardiac output, which is for average about 5 liters per minute. And so you may see that if you find this ratio between the supplied fresh air and supplied blood, it makes 0.8. This is average calculation. Why it's important? We need to understand that this ventilation provides a certain amount of oxygen. Blood that comes should get certain particular amount of oxygen to saturate blood, and there should be correspondence between these two amounts. And ventilation perfusion shows which ratio is normal, and we should maintain this ratio because otherwise there can be abnormal oxygen supply. And it's necessary to understand that it's only average ratio. In reality, in different parts of lungs, there is no the same value of this ratio. It's uneven. Due to gravity, both parameters, ventilation and perfusion, becomes high from lungs' upper lobes to medium and then to lower ones. Gravity contributes to greater blood supply to lower portions, and also even anatomy contributes to greater ventilation of lower lobes of the lungs. But nevertheless, it's so much uneven that because of perfusion, changes are much more intense. It creates the difference between upper and lower lobes in unexpected way. The maximal ventilation perfusion ratio is characteristic not for the lower lobes where both ventilation is high and blood supply is high, but ratio, the best one, is in the upper lobes. And ratio range is from as low as 0.7, which is actually in lower part of lungs, and up to even 3, which is characteristic for upper portions of the upper lobes. So this ratio influences the oxygen content of blood that leaves the pulmonary vessels. Of course, when all blood is mixed, it has normal characteristics, but each portion of lungs produces different saturation of oxygen, blood saturation with oxygen. For better understanding this, it's necessary to see the pictures and figures. Let's see. Physiologist West first divided lungs into the 3 zones according to difference in ratio between ventilation and perfusion. So, you see, from upper lobes to lower lobes, apex and base. Ventilation increases gradually from up to down direction. Similarly, blood flow increases, but increases much, much stronger. And now we can see on the figures, and now you will understand why this difference appears. Here we locate the lobes of lungs from upper to lower. Vertical axis will show both ventilation and perfusion. You see, ventilation increases from upper lobes to middle and lower. And perfusion also increases, but range of changes is much, much greater. Look. And so as a result, you see ratio is different and opposite. In the point of cross-section, ratio is 1, and it occurs somewhere between upper and middle lobes of lungs. Therefore, this is 1. For the lower lobes, you see, according to figures, simply it can be seen that blood perfusion is much greater than ventilation. So if you divide ventilation by blood perfusion, it makes less than 1. And it's about 0.7, actually. So for upper lobes, we have highest ratio, ventilation divided by perfusion. Then for middle lobes, it's a medium, and approximately it equals to average, 0.8, less than 1, 0.8. And for lower lobes, it makes about 0.7. Of course, when all blood that comes from the upper lobes, from middle and from lower, becomes mixed and goes through pulmonary veins back to the heart, the blood has normal characteristics, but initially maximal PO2 is characteristic for vessels passing through upper lobes, medium for middle lobes, and minimal PO2 for lower lobes. And when everything is mixed, it becomes of normal parameters. And for the standing person, we have the highest arterial PO2 above and in upper lobes, and lowest arterial PO2 in vessels of the lower lobes for a standing person. In a standing position, because gravity is the reason of differences in both ventilation and blood flow. In line position, difference between lobes that appear to be above, which are anterior initially, and the posterior lobes, which are now located in supine position. So difference is not such a great difference as we have in standing position. Therefore, we can consider that it's not of greatest importance. But in standing position, really difference of upper and lower lobes for both ventilation and blood supply is high. And you may understand that upper lobes have high arterial PO2, higher than normal, not 100 but more. As well as you may see that lower lobes have less than 100 millimeters mercury PO2 arterial. But blood that is mixed finally has its normal values about 100 millimeters mercury, with usual differences between 95 to 100 or really 105 and so on. So you should understand this difference of ventilation perfusion. Again, I remind you that average value is 0.8. And what happens in case of changes of this ratio? Let's consider this, the significance of this ratio. First, again I remind, ventilation provides certain amount of oxygen for saturation of blood. And perfusion, blood supply, provides certain volume of blood which should be oxygenated by this oxygen. Of course, between ventilation and perfusion there should be correspondence, because each portion of blood needs certain amount of oxygen. No more, because it cannot bind more than it can, but no less, of course. And most widespread types of abnormal VQ ratio comes because of decrease of something, like ventilation decrease or blood supply decrease. Let's consider first the case of higher ratio of ventilation perfusion. Usually it results from predominant decrease of blood supply. If perfusion decreases, ventilation appears to be higher than normal, so it brings more oxygen that can be bound by blood. So alveolar value of PO2 increases, PCO2 decreases because blood takes less oxygen that is supplied. As a result, nevertheless, arterial blood oxygen content does not become higher because hemoglobin under normal conditions already is saturated almost up to 100%. And really 100% are not achieved usually. So blood cannot take more oxygen than hemoglobin can bind. So as a result, part of alveolar space becomes ventilated uselessly, and this becomes a physiologic dead space. You remember that normally physiologic dead space does not differ much from anatomic, because in alveoli we don't have great amount of unused alveoli. But here now it becomes greater because not all the air from alveoli are used, not all the air. But as a result of all this, we have just unnecessary ventilation, and arterial blood quality remains normal. Arterial oxygen content, arterial PO2 is normal, as well as PCO2. But let's consider the opposite case, decrease of ventilation perfusion ratio. This results from predominant decrease of ventilation due to certain reasons. And now look what happens. Oxygen supply becomes not enough for oxygenation of blood until normal values. And as a result, both alveolar and arterial PO2 and PCO2 change, so that PO2 decreases, PCO2 increases, and we have hypercapnia and we have hypoxia. So this is a change which is dangerous, much more than previous one, because the arterial blood quality becomes bad, poor quality, and arterial blood does not bring to the body enough oxygen. This is a dangerous situation. Of course, both are abnormal, but second case is worse. Matching of the ventilation perfusion. We have certain mechanisms that provide, when possible, to match this, because the vessels of lungs respond to ventilation change respond to the decrease of ventilation by constriction. It's called hypoxic vasoconstriction. So It's unusual for the systemic circulation. This is entirely different. In the pulmonary blood vessels, when there is decreased oxygen in alveolar air, vessels become constricted. Let's recall what happens in the systemic circulation. Local factors are all vasodilating. Hypoxia produces vasodilation as well as hypercapnia. It's normal. When tissue suffers from insufficient oxygen and excessive CO2, it needs better blood supply, which can be achieved by dilation of vessels, and only then blood supply increases. But here, if the reaction will be the same as in systemic circulation, the alveoli with bad ventilation will get higher and higher blood supply. As a result, this blood will be even less saturated with oxygen, and there's nothing of useful for us. Actually, this is a protective reflex which helps to maintain, if possible, more or less better ventilation perfusion ratio and oxygen saturation, blood saturation with oxygen. So here notice that local factors produce opposite change. In the systemic vessels we have vasodilation in response to hypoxia, but in the lungs, in the whole pulmonary circulation, there is hypoxic vasoconstriction. If you remember these two words together, hypoxic vasoconstriction, will always able to answer what happens in case of decreased oxygen, decreased ventilation, and increased CO2 in alveolar air. The vessels of the lungs become constricted. And what is the use of this? Poor ventilation decrease, but if simultaneously we have decrease of blood supply, the ventilation perfusion ratio remains at least closer to normal, if not normal entirely. So, and if it's local hypoxia, it provides direction of blood from poorly ventilated areas into better ventilated parts of the lungs. And so it also permits to maintain better VQ ratio and arterial blood saturation with oxygen. As example, if there is local bronchial obstruction due to some reasons, it can be mechanical, for example, inhaled a foreign object, or it's really local bronchial obstruction because of the bronchial constriction, contraction of smooth muscles. In this area only, vasoconstriction occurs locally, and decrease in both ventilation and perfusion provides ratio still normal value, and arterial PO2 of blood that passes from this area remains normal. So much less blood, of course, goes out of pulmonary circulation, but the quality of this blood is not as poor as it could be without hypoxic vasoconstriction. Now let's consider the next stage, oxygen transport and later CO2 transport. For oxygen transport, the main thing you already know, that oxygen is transported by hemoglobin. As many other gases, oxygen is not well dissolved in blood, in fluids, and oxygen solubility is very low. And total amount per each 100 milliliters of blood of dissolved oxygen makes about 0.3 milliliters only. So in volume percent, milliliters per milliliters, it makes 0.3 volume percent. It's very low value. So we understand that simply by dissolved state, oxygen transport is not possible. We need much more oxygen, that only 0.3 per each 100 milliliters of blood. And it's necessary to bind oxygen. And also important that binding should be reversible. Bound oxygen should be easily given to the tissues when hemoglobin comes to the vessels, to capillaries of systemic circulation. And hemoglobin exactly meets all these requirements. This binding is reversible, and in lungs, hemoglobin easily binds oxygen. In the tissues, hemoglobin easily gives it back. So, for oxygen transport, therefore, we have two types with high predomination of main one, binding with hemoglobin. And also, it's necessary to remember that there certain portion, little portion, is dissolved. And exactly this dissolved portion creates partial oxygen pressure in blood, because bound oxygen cannot create pressure. Only dissolved portion is 0.3 milliliters dissolved in each 100 milliliters of blood, create this 100 millimeters mercury partial oxygen pressure of the arterial blood. As for binding by oxygen, something you know from biochemistry, but let's recall again. For adults, we have hemoglobin A, and these molecules consist of the 2 alpha chains and 2 beta chains, the 4 chains totally, and each chain contains the heme. Therefore, we have 4 hemes. Heme is the key portion of the big molecule, which is oxygen binding part. And binding occurs exactly in the area where we have a ferrum atom of iron. Iron is 2+ valency and does not become changed by binding of oxygen. It remains 2+, no 3+, normally. Therefore, with 4 hemes, a hemoglobin molecule, each molecule can bind 4 oxygen molecules. The binding occurs not equally. First molecule is bound less easily, then second easier, and third easy, and so fourth molecule is bound easiest and fastest. And when all 4 molecules are bound by each heme correspondingly, this substance is oxygenated hemoglobin, or simply in short, oxyhemoglobin. When at least one molecule is released of oxygen, so this is already deoxygenated, regardless of 3 molecules remain 1, or 3 molecules 2, or 1, or none. Usually not all molecules become dissociated. Oxygen capacity of blood. Generally, of course, it depends on hemoglobin content. The more hemoglobin is present in each liter of blood, the more oxygen this blood can bind. So oxygen capacity is directly proportional to the hemoglobin. And standard value, you should know, 1 gram of hemoglobin in the body binds 1.34 milliliters of oxygen. You can find some higher value like 1.39, but this is not for body conditions, it's a theoretical calculation. But in reality, it binds 1.34. So if we take the standard value of hemoglobin as 150 grams per each liter, we can calculate 1 1/3 approximately multiplied by 150. It makes about 200 milliliters of oxygen per each liter of arterial blood. Of course, if the person has different value of hemoglobin, we can easily calculate yourself for any other value like 120 or 130, or sometimes it can be higher, 160 or so, you can calculate. But average content considered to be 200 milliliters per liter of blood, or if it's expressed in volume percent, you remember percent is per 100 milliliters. We have correspondingly not 200 milliliters of oxygen, but 20 milliliters. And so it makes 20 volume percent. Volume percent means milliliters in milliliters, because simply percent are gram in milliliters, in 100 milliliters. So 20 milliliters of oxygen per 100 milliliters of blood. So actually it's a big amount. You see, arterial blood in each 100 milliliters carries 20 milliliters of oxygen as a result of binding by hemoglobin. And we understand hemoglobin content is fixed. In blood, oxygen capacity is also fixed. And so when we start some physical activity, for example, we cannot change the oxygen capacity. And therefore, the way to deliver more oxygen to the body is to increase the number of liters of blood delivered each minute to the body, or simply cardiac output. So oxygen requirements increase, stimulates the heart, and heart increases the cardiac output, so that more liters pass through the cycle per each minute and more oxygen can be supplied. But each liter contains the same usual amount of oxygen. So also the dissolved portion of oxygen is fixed under normal conditions. We can increase the amount of dissolved oxygen by creation of higher than atmospheric pressure. But significantly higher pressure can be created in the barometric chambers. Or naturally it may occur if there is underwater submersion. But it's not usual case. So under normal conditions, the dissolved portion remains low as usual and with regular atmospheric pressure. Then oxygen binding by hemoglobin. This is very important part of the whole theme. Ability is variable for binding because, remember, hemoglobin should be able to bind oxygen, but simultaneously it should be also able to release it easily, to dissociate. So variable affinity is expressed by oxyhemoglobin dissociation curve. This is the specific figure which we are going to consider in many details. And this is a plot figure of oxyhemoglobin percentage depending on the blood partial oxygen pressure. So the horizontal line axis will contain PO2 of blood, and vertical axis shows percentage of oxygenated hemoglobin out of the total hemoglobin available. Maximal oxygen binding by hemoglobin occurs at the highest level of PO2, which is normally 100 millimeters mercury. Alveolar and arterial blood PO2 level. So when blood passes through alveoli, it has PO2 pressure is 100 millimeters mercury, then hemoglobin binds oxygen maximally. But when PO2 becomes less, as it occurs when blood enters the systemic arteries, arterioles, and especially capillaries, oxyhemoglobin content decreases because oxyhemoglobin dissociates and releases oxygen as follows. Hemoglobin O2 becomes converted to hemoglobin separately and oxygen molecules separately, and these oxygen molecules can diffuse into tissues and go into cells. This relationship is nonlinear. It could be very easy to predict level when it's exactly proportional, but it's nonlinear. The curve has a sigmoid shape due to increase of oxygen affinity after binding of every next oxygen molecules. As it was already said, the first molecule binds not so easy as next one, and each next binds easier and easier, with maximal affinity for the last fourth molecule. And similarly, the release occurs. First molecule not easy, then easier and easier. It becomes to release molecules of oxygen from oxygenated hemoglobin. Let's do this dissociation curve. You should be able to draw this figure yourself, and for this you need to know at least 2 points additionally to the upper and lower points. So it's simply square because we have 100 millimeters mercury of PO2 on the horizontal axis, and we have 100% of hemoglobin totally. So 100 per 100, it makes perfect square. And starting point is 0, of course, because no oxygen, no oxygenated hemoglobin. And final point to the right upper angle is not perfectly 100, but about this. Starting is 0 and maximal is 96-98% of oxyhemoglobin for the arterial blood. Theoretically, if a relationship is direct, straight, it could be such a line. But as you remember, it's not true. We have the sigmoid shape. Let's for comparison, we leave this line, but let's place exact values. And additionally to this 0 point initial and 96-98 maximal, we need to remember at least 2 values. For 40 millimeters mercury, the corresponding point is not 40 as it could be, but 75%. So this is one point to remember, 40 and 75, a pair of values. And then 60 and 90%, 60 millimeters PO2 and 90% of oxygenated hemoglobin. So with these 2 points already you can produce normal dissociation curve of oxygen. But the best case if you know also the value that corresponds to 50%. This is so-called index P50, about 25 millimeters. To be more exact, it can be 27, but 25 is also not abnormal. So 27 millimeters, it is P50, the pressure of oxygen which provides 50% of oxygenated hemoglobin. With these 3 additional points, you can easily produce the dissociation curve. And if it goes from 0, it considers the process of saturation of hemoglobin with oxygen. really we call this curve dissociation curve, and for consideration of the process of dissociation we should start from the point where there is no dissociation, where oxyhemoglobin content is maximal. So from upper point we should start and go down. And notice that what is the advantage of this sigmoid shape of the curve? That blood, even 60 millimeters PO2, which is really hypoxia, provides already saturation of blood with 90% of oxyhemoglobin. So basically it shows that we can withstand hypoxia even until 60 millimeters PO2 low. You see, it is 40% less value than normal, and still oxyhemoglobin content decreases only less than 10%. So we can withstand the hypoxia until certain degree. And in case of direct relationship like this straight line shows, instead of 90 we can get only about 60. So it could be much more serious problem. It will be severe hypoxia. So we are protected from very high sensitivity to hypoxia due to such a shape of oxyhemoglobin saturation dissociation curve. Now, if we consider really dissociation, we start from the upper point and maximal oxyhemoglobin content, and then we go downward. This is dissociation, but you may see that usually the dissociation curve does not go to zero level. Actually, hemoglobin practically almost never remains without oxygen at all. Look at the values of PO2 and the horizontal axis. This 100 is corresponding to arterial blood PO2, and venous blood PO2 is 40. So you see, generally we use only this range. For average blood, we use only this range of PO2 values, from 100 of arterial till 40 of venous. Then venous blood goes into lungs, and from 40 it turns back to 100. Correspondingly, the range of values of oxyhemoglobin content varies from almost 100 till 75. So normally venous blood contains 75% of oxygen initially present in arterial blood. So 3/4 of oxygen still remain in blood. Of course, it's for resting state, because when oxygen requirement increase, we use more oxygen and remaining amount of oxyhemoglobin decreases. But this is our additional storage of oxygen, and we can use more. But resting state does not require such a great amount of oxygen, and we have oxyhemoglobin in venous blood 75%. And actually, dissociation curve, saturation goes, when venous blood comes into lungs, saturation goes like this, from 75 up to almost 100. And when blood goes into systemic capillaries, oppositely, from almost 100 it comes back to the 75%. Of course, it's for average, when it's mixed venous blood. You see, therefore, our blood, even venous blood, contains 75% of oxyhemoglobin, and this is remaining oxygenated hemoglobin. Therefore, the upper portion is oxyhemoglobin that was dissociated into hemoglobin separately and oxygen. So exactly this amount of oxygen is used. So you may see that this is oxygen utilization quotient, actually 25% at rest. But when our requirement in oxygen increases and body cells take more oxygen, PO2 of venous blood moves to the lower values. You see what happens to oxyhemoglobin. It quickly starts to dissociate and releases much more oxygen than usual. And the curve goes down much faster than before. So it's our reserves for better oxygen delivery in case of physical activity. And also we have additional mechanisms to provide more oxygen for tissue cells, because these points and this type of curve exactly characteristic for normal conditions such as CO2, pH, temperature, and also the force index here is the concentration, square brackets mean concentration, and inside is 2.3 diphosphoglycerate. This substance is produced by metabolic processes in red blood cells, and normally its concentration is very low, and so it provides normal curve dissociation. But in case of hypoxia, metabolic processes produce more diphosphoglycerate, so it can influence the dissociation curve position. The position of this curve is normal when all parameters are normal. Our PCO2 of arterial blood is normally 40 millimeters, pH with plus minus but average is 7.4, temperature in the lungs is 37, and in the body average in core also. And concentration of diphosphoglycerate, 2.3 diphosphoglycerate, is normal. So this curve goes like this. But under conditions of high activity, we have change of all these parameters. We may have. This was designed for case when lecture was shown on the screen, but now also we can use this stage as normal curve, because later we are going to discuss shifts of the curve. Look, when oxygen requirements increase, in case of physical activity mostly, but also it can be emotional stress, but mostly, of course, physical activity. Look, the dissociation increases. Body needs more oxygen. Therefore, oxyhemoglobin gives more oxygen to the body, and remaining amount of oxyhemoglobin decreases. You see points go downward on the vertical lines. And new curve, it starts again from 0, of course, and in maximal value of PO2 it comes to normal values. So only middle portion of the curve shifts, but look what kind of shift occurs. Really the curve appears to be positioned to the right, and really we call this right, like this shift, the shift to the right. So such a shift to the right occurs when oxygen requirement increases and parameters shown in the box to the left become changed to the side of higher CO2, less pH, but it corresponds to higher H plus ions, higher temperature, higher diphosphoglycerate concentration. Each of these changes produces shift to the curve, and correspondingly all the changes simultaneously produce greater shift to the curve. The case of a shift to the right shown now is for example. It can be stronger shift, it can be less shift, so it depends on situation. But in any case, oxyhemoglobin content decreases and dissociated portion increases. For this particular picture, it makes about 50% dissociation as compared to initially 25% of dissociation. You see, much greater oxygen delivery to the tissues occurs when oxygen requirement increase because of activity. And physical activity produces these changes. PCO2 increases, H plus concentration increases, temperature itself, if temperature increases, you know that all metabolic needs become higher. And so oxygen delivery becomes not enough, and so it increases the requirement in oxygen. And about 2.3 diphosphoglycerate, I already told that it's produced under hypoxia. So amount, it's directly proportional to the degree of hypoxia. And all these factors produce shift to the right. Shift to the right, you see? But notice, if you use instead of H plus concentration, if you use pH, the arrow direction falls out of the general rules. Actually, the factor that changes, that influences the hemoglobin molecule is not pH logarithmic calculated index, but H plus ions themselves. So therefore, it's more logical to use H plus, and in this case, it's easier to memorize the 4 factors that produce the shift to the curve. If all these 4 factors, or even one at least from these listed factors, changes upward, it moves the dissociation to the right. What it means? Dissociation shift to the right means that dissociation increases. Therefore, remaining amount of oxyhemoglobin decreases. Look, shift to the right is the same as decreased affinity. Hemoglobin takes less oxygen, keeps less oxygen, and easily gives it, so dissociation increases. Affinity, when molecule takes oxygen, grabs it, and does not give to anyone, it keeps to itself. But decreased affinity means easy giving to the blood, easy dissociation. So we see shifts to the right, decreased affinity, and increased dissociation. All are like synonyms here. So, and now we have also possible shift to the opposite side, shift to the left. Factors that may produce this shift to the left are all factors directed downward. When there is a decreased PCO2, decreased H+, or higher pH, which is alkalosis, when there is a decrease of temperature, hypothermia, and decreased concentration of 2.3 diphosphoglycerate, the dissociation becomes less because of higher hemoglobin affinity to oxygen. Hemoglobin does not give oxygen and keeps bound to hemoglobin. In this case, the percentage of oxyhemoglobin remains high, and points on the vertical lines go upward. Less dissociation, more remaining oxygenated hemoglobin. So percentage of oxygenated hemoglobin becomes higher. Degree is variable of this increase, but here, for example, you have 3 points changing, and curve that passes through these points become shifted exactly to the left. So you understand, shift to the left means less oxygen given to tissues. Blood contains this oxygen, but hemoglobin doesn't give this oxygen. And normally it corresponds to less oxygen requirements of the tissues. For example, less temperature of the body, hypothermia. It decreases oxygen dissociation, but actually when temperature is decreased, the requirement in oxygen is also decreased. So dissociation, you see how much less becomes as compared to normal. And this is not necessarily photo, but you can make print screen, and you see the all shifts shown here. Then pay attention to the index P50. Actually, for studying purposes, you should know that values such as pair of values 40 to 75% and 60 to 90%. But clinical estimation of the shifts usually is done on the basis of the P50 value. Normally, as you remember, 25-27 millimeters mercury. And conveniently, the point of P50 moves to the same direction where the curve goes. You see, for the hour curve with shift to the right, value of P50 appears to be exactly 40 millimeters. So P50 from 25 moved to the side to the right, to the 40 millimeters. Then, for the shifted to the left curve, look, this value of P50 becomes less now. It's about 16 or so if you compare. So P50 decrease shows that shift to the left. P50 increase shows shift to the right. So movement of the point occurs with the same direction with the curve. Because you see, cross-section of the curve with the horizontal line 50% always moves to the same direction where the curve itself goes. So if you know P50 norm, you can always predict it. On the basis of P50, you can make conclusion if there is a shift to the left or shift to the right. Of course, more widespread is shift to the right, because situation of hypocapnia, alkalosis, decreased temperature, as not as often as opposite situation of hypercapnia, acidosis, and high temperature. But all this happens. Now, oxygen release correlation to oxygen utilization. All that we considered on the curve is refers to the mixed venous blood. But blood passes through different organs, different tissues with different oxygen requirements. So value of venous blood at rest is 75% of oxyhemoglobin. But it's only after mixing, because you remember that we have different organs that have very different oxygen requirements. And let's observe the minimal and maximal values of oxygen utilization. Look, with high oxygen utilization, there will be greater decrease of PO2 in both interstitium of the tissue and venous PO2 value. And best example of the organ with high oxygen utilization, you still remember because it was not long ago, we considered the heart with its very high level of oxygen usage. So here you see what happens. Venous PO2 is less than usual because oxygen cells take more oxygen than many other tissues. Therefore, it can be as low as less than P50 even. So you can observe dissociation. It goes down until reaching the corresponding value. So there is very little amount of remaining oxyhemoglobin and high amount of dissociated hemoglobin or dissociated oxygen. Look, if initially it's about 100, really not 100, minus 35, for example, it makes 65. So this is exactly the amount which is used by the heart. And you remember, heart utilization coefficient is between 60 to 70 at rest. So for our approximate figure, 65, it corresponds to normal. And on the basis of this, you can see that PO2, venous PO2, is even less than 25 in the heart vessels because of high oxygen usage by the heart, high utilization. So you see, blood that comes out of veins of the heart contains less than many other tissues, less oxygen, less oxygenated hemoglobin. Not 3/4 like 75%, but less than 40 or even about 35%. So it means that each hemoglobin molecule, oxyhemoglobin molecule, gives more than half, or about 3 molecules of oxygen out of each oxyhemoglobin. Because heart needs it, and hemoglobin provides, releases. So the poor content of oxygen is characteristic for venous blood that goes through venous vessels of the heart. Opposite case with low oxygen utilization we can consider as example the skin. Skin cells, you remember, do not have great oxygen requirement, they don't have great metabolic rate, and they therefore do not decrease PO2 much. So in venous vessels, PO2 is closer to arterial, not exactly equal, but closer. Venous PO2 is higher than many other tissues. For example, let's take 60 millimeters. And you see, therefore, dissociation of hemoglobin occurs only until value, if we choose this 60 millimeters, until 90% remaining oxyhemoglobin. So dissociation itself makes less than 10. 100 is certain rounded value, really it's less than 100. So it's practically less than 10% of oxyhemoglobin dissociates. And not from each molecule even, we have release of 1 oxygen. And oxygen coefficient is very low here. So you may understand why the blood produces, for white skin, produces a pinkish color of skin, because the skin color determined not only by melanin, of course, but additionally is determined by color of venous blood, because venules are easier seen than arterials, and they're seen through the epithelium. And so, due to venous blood color that does not differ much from arterial, the blood with the skin of people with white skin look like pink colored, because color is not bluish. Bluish color it's characteristic for the cyanosis, for hypoxia case. With normal case, we have normal blood, venous blood color very close to the arterial. So, and you remember, when all blood becomes mixed in the right atrium, the mixed venous blood, it makes a usual 40 millimeters mercury at rest, because we combine all. Some molecules of hemoglobin lost oxygen, much more than usual. Some molecules lost almost, not all the molecules even released hemoglobin, released oxygen like in skin, and so totally it makes average value. And average oxygen utilization coefficient is about 25, or depending on individual characteristics, up to 30%. But standard, according to figure, which we considered previous slide, is 25 for resting state. Let's consider next important moment, the carbon monoxide poisoning. Normally, hemoglobin is designed to bind oxygen, but there is a substance that binds much easier to hemoglobin, and this is CO, carbon monoxide. Affinity is much greater of hemoglobin to CO, and therefore, if there is a little amount of CO molecules in the air, the hemoglobin will predominantly bound to CO. And if one molecule of CO is attached to hemoglobin, so the remaining oxygen molecules become very hard to release. and dissociation becomes decreased, practically it's shift to the left. Look again, this is normal, oxygenate, not still oxygenated, but only 3 molecules of oxygen are present, and the 4th molecule is expected, but CO binds, it binds instead. So we say this is carboxy hemoglobin. Notice, carb hemoglobin is hemoglobin bound to CO2, carb hemoglobin or carbohemoglobin. But if there is oxy between, this is pathological carboxyhemoglobin bound to carbon monoxide, CO. And the remaining oxygen molecules, this hemoglobin will hardly release. Therefore, the body has hypoxia. Blood circulates, but oxygen is not given. Look at the dissociation curve. Dissociation curve of the carboxyhemoglobin. You see, it looks like the hemoglobin dissociation curve, but the values on the horizontal axis, you see, just 0.4 millimeters mercury is enough to saturate all the hemoglobin fully by carbon monoxide. So you may understand that little amount of CO is enough to have in the inhaled air to produce serious poisoning. Just 0.1% of carbon monoxide in the air brings to hemoglobin CO about 80% of already. So it's severe hypoxia because remaining 20% will not be enough to produce delivery of oxygen to the body. In these cases, the help should come as soon as possible. With best type of help is breathing by pure oxygen, which increases the rate of CO release. Pure oxygen makes it faster to release CO, and finally oxygen takes its place and hemoglobin again becomes oxygenated, oxyhemoglobin. In case of not available pure oxygen, at least fresh air should be supplied as soon as possible to stop further additional CO presence. Then we switch to the transport of the carbon monoxide. Carbon dioxide. Monoxide was bad poisoning. Carbon dioxide, CO2. It is formed in the tissue cells and it goes into interstitium, finally to blood plasma, and transported in 3 forms. Oxygen is transported in 2 forms with hemoglobin. But for CO2 we have 3 forms of transportation. Let's see. Let's list all these 3 forms. Oxygen goes into mitochondria and is used for the respiratory chain to produce energy, and as a result CO2 is formed as well as water. This CO2 partially becomes simply dissolved in plasma. Not great amount, but as compared to oxygen, more CO2 is dissolved because, if you remember, this solubility coefficient is about 24 times better. So it's simply dissolved. And from all the CO2 formed, it's about 5%. So we started from a minimal type of transportation, minimal way. Then, second case is formation of carbaminohemoglobin. So it binds to hemoglobin, but not to heme, of course, to protein portion, where amino acids bind, amino groups in amino acids bind this CO2. You see NH2 plus CO2 form the NH, then COOH, carboxyl group. And so this is carbaminohemoglobin. And these amino acids carry COO converted to carboxyl groups. And this transport makes about 15% of all transportation. You see, 80% remains for the main way of transport. And this main way is formation of HCO3 minus ions. You see, CO2 is bound with water that forms the carbonic acid, and carbonic acid dissociates to H plus and HCO3 minus. And this HCO3 minus, or bicarbonate ion, is the most important way of CO2 transport. It makes about 80%. And it's important to understand that formation of carbonic acid from carbon dioxide and water, the reaction that goes very slowly naturally. It's not easy to make the CO2 become converted to H plus and H2O3 minus. But this reaction is accelerated many times by the enzyme which is called carbonic anhydrase. And this enzyme is present inside red blood cells. So to be converted to HCO3 minus ion, molecules of CO2 should enter red blood cells. And they readily enter because the concentration of CO2 in red blood cells is constantly zero. You see, each molecule of CO2 that enters red cell immediately becomes converted to ions, and so CO2 content is zero. So it constantly drives more and more CO2 molecules to enter the red cells. Carbonic anhydrase is present inside. So you may remember now that not only hemoglobin is contained inside the red cells, but also carbonic anhydrase, protein that is enzyme. And main way of transportation, I believe you remember, HCO3 minus. Let's consider how this carbonic anhydrase works, because it can produce reaction in both directions. In tissues reaction we already considered, but let's consider again. There is hemoglobin, there is carbonic anhydrase, and CO2. CO2 enters the red cells. When CO2 enters, immediately carbonic anhydrase produces the reaction of formation of carbonic acid, and H plus ions are formed as well as HCO3 minus. HCO3 minus anion is moved out of red cells into blood plasma in exchange with chlorine. This is specific exchanger with chlorine. And H plus ions, which can create serious acidification of blood. These H plus ions are bound by hemoglobin. Chlorine entry is called chloride shift. CO2 is zero, and H plus bound by hemoglobin. So we call this the buffering function of hemoglobin. You see, hemoglobin provides not only one function of oxygen transport, but also it prevents great acidity of the venous blood, because many ions, H plus ions, become bound to hemoglobin temporarily until the moment of reverse reaction, and therefore they don't affect the total pH of the blood. Reactions in the lungs are opposite. When red cells with blood enter the pulmonary capillaries, here we have hemoglobin in its H plus bound state. Then we have carbonic anhydrase as usual, and we have HCO3 minus ion transported in blood plasma. And now again we have opposite movement. HCO3 minus moves into red cells in exchange with chlorine. HCO3 constantly will move there because it disappears again. It becomes converted. The movement of HCO3 minus in exchange with chlorine is called reverse chloride shift. HCO3 minus becomes bound with H plus. Again, let's observe. In the moment when hemoglobin binds oxygen, and you remember it binds oxygen because oxygen level here is high. According to Dissociation Curve, 100 millimeters mercury of oxygen makes hemoglobin readily bind oxygen. In the moment when oxygen is bound to hemoglobin, H plus ions are released, and then carbonic anhydrase quickly convert HCO3 minus and H plus into carbonic acid. And now carbonic acid becomes broken into CO2 and water. CO2 goes into plasma, and from plasma it diffuses easily into alveolar air, where CO2 content is less. And so this reaction occurs easily. Transport of bicarbonate and chloride ions is performed by a special bicarbonate chloride carrier protein, which is present in membrane of red blood cells. And I remind you that carbonic anhydrase increases the rate of reactions 5,000 times. Diffusion of gases in the tissues. we consider the processes how transportation occurs, and in tissues diffusion again occurs down the gradients, so that it's enough just to put values of pressures, partial pressures for both gases. But diffusion occurs here into stages. First, from blood, the gases, oxygen in particular, should move into interstitium, and from interstitium it should move into cells. And CO2 should move in the opposite direction. So PO2 values of arterial blood are about 100, and PCO2 is about 40. Interstitium should have, if you don't remember even, you can predict, interstitium should have less PO2 and higher PCO2, and cells corresponding even less PO2 and higher PCO2. So here is from 20 to 40. Upper limit should be not higher than venous value of PO2, and venous value is 40. Cellular value corresponding is almost up to 0, but it's variable depending on intensity of metabolism in the cell. And lowest value of oxygen is characteristic for mitochondria, where oxygen is used. So, and you see, gradient is directed from higher to less and less pressure. Finally, oxygen goes into mitochondria, where it's used immediately. As a result, CO2 is produced, as well as water. Constant diffusion of oxygen to the cells is driven by constant oxygen consumption in the tissues. Constant, I mean that it goes without any stops, but really level of consumption is not constant. It can be variable, depending on intensity of metabolism. And so you may see when metabolism becomes more intensive, it uses more oxygen, and level of PO2 in cells becomes less. The less is this level, the higher difference exists between arterial blood and cells. So gradient increases, and it provides much more oxygen delivery to cells. So exactly this is a driving factor for the diffusion of oxygen, intensity of metabolism. And CO2 also correspondingly is produced in proportion to intensity of metabolism. And from highest level in the mitochondria, CO2 easily moves into a cell, cytoplasm, and then to interstitium, and finally to arterial blood. The minimal level in interstitium should be no less than in venous blood, which is 46 value. Then it should be higher and higher progressively, and simply value maximal value 80 must be remembered. So for CO2 goes from mitochondria into cytoplasm, where level is less, then to interstitium, where level is even less, and finally it goes into blood. And final value for the blood, which becomes converted to venous, is 46, as you remember, for resting state. So you may see that rate of tissue metabolism determines the rate of oxygen delivery. Arterial blood normally has standard values of PO2 and PCO2, but cell, you see, have variable PO2 and PCO2, and the greater is intensity of metabolism, the less is PO2, the higher is PCO2, and both changes increase the gradient for gases. Oxygen utilization coefficient. We already talked about this, but now let's calculate it exactly. It's a part of oxygen utilized from the arterial blood. Oxygen utilization quotient, therefore, or coefficient, is difference between arterial and venous oxygen content, which is used oxygen. Also, it's called arterio-venous difference, and divided by initial content in arterial blood. And how we can calculate it? First, we can use the volume percentage of oxygen in blood. We already calculated that for standard blood with 150 grams per liter of hemoglobin, oxygen content, according to oxygen capacity, makes 20 volume percent, which is 20 milliliters per 100 milliliter of blood. Then, resting venous level of PO2 is 40, and it corresponds to 75% of oxyhemoglobin. And 75% is 3/4 of initial. And so if initially is 20, now it's 15 volume percent. Let's recall this. Normally, blood dissociation, oxyhemoglobin in blood dissociates from almost 100 till 75% of oxyhemoglobin. And so this is remaining oxygenated hemoglobin, and the difference is about 25. Let's calculate this difference. 20 volume percent initially, and remaining is 15 volume percent. So it makes 5 volume percent utilized. And divided by initial 20, it makes 25%. So standard resting value of oxygen utilization coefficient is 25%. But you remember that for different organs it can be different. Let's recall the values of coefficient for the body as a whole and for different tissues. It's enough to make 2 organs and the body as a whole. The body as a whole, then skeletal muscles for comparison, and the heart. And 2 values should be for each, resting value and maximal value. For the body as a whole, the values coincide with skeletal muscles, 24 to 30 at rest. And maximally it can go up to, for the heart, you remember it's higher than usual. Even at rest it makes from 60 to 70%. While 60% is maximal, which can be achieved by the body as a whole at maximal activity, and skeletal muscles separately also. So values coincide, perhaps because skeletal muscle make big part of the body as a whole and have great variety of the values of oxygen requirement. And the heart, as you also remember, may use at 70 or even 80% of oxygen when there is a maximal physical activity. So these values we already started, but now let's recall them and put together all. This is main portion of the lecture, and thank you for your attention. Hope you'll study all this successfully.